So here's your video, the second video for section 18.3 on genetic recombination in bacteria. And this time our source of genetic recombination is going to be something called transduction. And transduction, what is happening here is we're going to have a virus aiding in the carrying of some genetic information from one cell to another cell. Okay, transduction uses a virus. So our diagram here explains it all, as they usually do. Here we have a bacterial cell going through, excuse me, we have a virus injecting its genetic material, and its DNA in this case, going through the typical viral steps. And so the DNA of the host is getting broken apart, and then we go through the whole process of making proteins and viral DNA and all that. We assemble some of the viruses, and here's where the mistake happens if I see this A plus here. Okay, what we're having is genetic information from this first bacteria is accidentally being put inside of the virus. Okay, and so instead of having viral DNA, it has host DNA. The host DNA being in there means that this will no longer be able to function as a virus as far as taking over a cell and making more viral DNA and all that. But what it still can do is it can attach to a new bacteria. It can insert the DNA that it has, and this time instead of inserting viral DNA, it's going to be inserting DNA from, we're calling this the positive bacteria up here. It has the A plus gene and the B plus gene, doesn't matter. But it's inserting the A plus gene segment from the original bacteria. Now, what I like about this picture is this is showing the crossing over idea, and I've mentioned this in previous videos where I said through a mechanism similar to crossing over, some genetic material would be transferred from one place to another. And this is actually showing it. So what has to happen for this this crossing over action to happen is we have to have whatever the new DNA is. In this case, it's from the positive bacteria. It has to match up with the same location on the chromosome of the second bacteria, the negative bacteria here. And so if they line up perfectly, then crossing over can happen where we basically just swap the material. So the positive A ends up in this genome where there used to be a negative A. Okay, so we get a new combination of DNA, and it was a virus that helped with this. Okay, so that's a genetic recombination through transduction, and then now we'll move on to the next type. All right, our next type of genetic recombination, is something called conjugation. Conjugation is as close to sexual reproduction as a bacteria is going to get. So sometimes they do call that, they do refer to this as sexual reproduction or recombination within bacteria. But anyway, this slide is just going to be some definitions. Basically, with conjugation, we're going to have two bacteria directly linking to each other. And after they've linked to each other, we're going to have some DNA or genetic material transferring from one to the other. Excuse me. This diagram here is showing something called a sex pilus, which is the singular, plural is sex pili. But basically, excuse me, sex pili is a appendage that can go, and I don't know which one is the donor bacteria here, but that can basically reach from one bacteria to another. And we'll say that this one reached out to this one, connects, and then it'll draw the two bacteria together. Okay, so that's what a sex pilus does, draws two bacteria together. Once they're together, they develop something, so that was a sex pilus. They develop something called a mating bridge, and if I were to draw two bacteria that are made a mating bridge, it's basically a connection between their cytoplasms. That's horrible. There we go. Between their cytoplasms so that genetic material can go from one to the other. So that's a mating bridge. We'll see a diagram of this on the next slide. Other terms that you'll see, something called the F factor, F standing for fertility. The F factor is basically the genetic sequence that codes for the ability to make a sex pilus. Okay, that's what the F factor is. And so if this bacteria here was the one that made the sex pilus, then it would contain the F factor, which allows it to do this whole conjugation thing. Another term that's just being thrown in here is a plasmid, because we're going to start to talk about plasmids in a moment. A plasmid would be, if you remember, a bacterial cell has its chromosome. So there's a chromosome. A plasmid is an extra set of DNA. It's circular as well, but it's an extra set of DNA that is not the genome of the bacteria originally, and it carries extra genes on it. So there is a plasmid called the F plasmid we'll learn about, and that F plasmid actually carries the F factor, okay, the genetic information for making the sex pilus. Okay, so that's a plasmid. And then finally an episome, okay, or an episome. An episome is something that it's genetic material that can either be replicated by itself or can be inserted into the host DNA and replicated. A plasmid is an example. Here, this plasmid, when it goes to replication, it could replicate independently by itself, okay, or it could actually insert itself into the host genome, and then when the host genome, when the genome replicates, it would be replicated along with it. That's what makes it an episome. Okay. Other examples of episomes would be if you look at viruses that can do the lysogenic cycle. They call them temperate viruses if they can do both the lysogenic and lytic. That DNA either inserts itself into the host and is replicated, or it can actually replicate itself if it goes right to a lytic cycle. Alright, so that's the intro to conjugation. Now we're going to look at some better diagrams. Here is our better diagram, if you will, or specific diagram of conjugation. And so we have two cells here that are already connected by a sex pilus would have already connected, and we have a mating bridge already formed here, mating bridge. And if you remember, in order for that sex pilus and the mating bridge to be formed, one of the cells had to have an F factor. Sometimes the F factor is carried in a plasmid. That is then called the F plasmid. And we're having this here. And what we're showing is conjugation of the F plasmid from one bacteria to another. So because the F factor is present, the two can form a mating bridge. When it is the F plasmid that is transferred from one side to the other, what we have happening here is a special type of replication, so that at the end we're going to see that we have two copies of the F plasmid. What ends up happening here, and this diagram really copied poorly, but the original plasmid is in dark blue, and what we're having here is basically the plasmid is separating its strands so that one of the dark blue strands is coming across into the second bacteria, and then one of the dark blue strand is staying here. As the one strand crosses into the new bacteria, replication happens, and so you can see a light blue line showing up beside this, making it double-stranded. Replication happens here, and replication completes over here as well, where we make a light blue copy to go along with the original dark blue copy for the DNA, so that in the end we have like a semi-conservative replication situation going on between the two, where each has part old and part new DNA. Anyway, but we have gotten this genetic information from one bacteria into another bacteria, and then the mating bridge is going to break, and we've had conjugation happening. So this is one type of conjugation. This is basically all it's really doing is giving the ability to make the sex pilus, just spreading that ability from one bacteria to another. So it's not a lot of recombination. There's not other genes involved here. But we're going to look at another type of conjugation quickly that does start to move some of the genome of the bacteria from one to another. Okay, another type of conjugation here that's going to give us a little bit more bacterial recombination is when our F plasmid, so here's our F plasmid, sometimes the F plasmid will insert itself into the genome of the bacteria, the host genome. So here we have it. When this happens, we call it an HFR cell, or high frequency of recombination cell, because it has this F factor inside of its host genome. So that means that if we go down here, that F factor coded for the ability to make the sex pilus and the mating bridge. So that has happened here, and the F factor is going to do exactly what it did when it was part of a plasmid, when it was the F plasmid, except that now, as it breaks apart and starts to go through its replication or whatever, sometimes it will take some of the original host genome with it. Okay, so we have our the dark blue and light blue here, like we had before, but now some of the host genome is kind of tagging along behind it. So once the conjugation is done, I have host genome as well as my plasmid genome, the F factor genome. I have those both into this new cell, the recipient. And again, I like this diagram because it starts to show some crossing over that can happen. So it's labeled genes, and if the B genes happen to align up, here the B genes have happened to align up, we can have crossing over happen where we have the genome of the recipient cell combined with the genome of the donating cell here, and then the rest of this DNA maybe gets chopped up by the bacteria, because bacteria do have the ability to chop up extra DNA that doesn't belong. But anyway, now this is legitimate genetic recombination. We're taking some of the genome of one cell and adding it into the genome of a different cell, and this can cause a lot of variation within bacteria. Okay, so that's conjugation that way. The other thing that I'm just going to introduce here is with this F plasmid that we've been talking about. The F plasmid is a plasmid that has that F factor in it, so it's going to be more fertile. There are other types of plasmids, and one that we're going to talk about a lot this year is called the R plasmid. The R in this case stands for resistance. And I guess I can write plasmid here, and it will be resistant. I didn't have to do that. Anyway, it'll be resistant to antibiotics. Okay, so there are plasmids that exist that are resistant to antibiotics, and some of those plasmids also contain the F factor, which means that they are resistant to antibiotics and they can pass that to another cell through conjugation. And we're going to look at some experiments that have had that as well in the future. So, okay, that's it with conjugation. We got a couple minutes left here, and we're going to talk about recombination within a bacteria. Okay, our last little bit of recombination that's going to be happening, or that we're going to talk about, is something called transposition. This is not between bacteria, it's within one bacterial cell, or wherever the transposition is happening, it's within that one cell. There's different ways this can happen, but basically it's taking a segment of DNA and moving it from one location in the genome to a different location in the genome. And this can either happen as a cut and paste, meaning we cut the sequence out of the genome and then paste it back in somewhere else, or it could be a copy and paste where we make a copy of the sequence and then paste the copy in somewhere else. We're going to kind of just look at the cut and paste variety here, but the first term or thing to know here is something called an insertion sequence. An insertion transposition basically takes a segment that has the gene for something called transposase, which is basically the enzyme that does the transpositions. So it has that enzyme gene, and then on either side of the enzyme's gene, it's going to be something called an inverted repeat. And inverted repeats basically means upside down and backwards is inverted. So here I've got ATC, CGCT. I've got the same thing over here, ATC, CCGGT, whatever. If I look at it upside down and backwards, this and this are exactly the same. Okay, so that's the inverted repeat. Essentially, transposase has the ability to recognize, that enzyme can recognize these inverted repeats, and can basically go through the process of chopping these away and then inserting them somewhere else. The whole process there doesn't actually involve the cutting out of this and then carrying it to somewhere else and removing it. The enzyme actually brings the two locations together so that as this is cut, it's immediately inserted to the new location without being like carried there. Okay, so that's how a transposition of an insertion sequence happens. The other type of transposition has to do with a transposon. A transposon, or sometimes they call these jumping genes. And a transposon, it's the exact same process. The difference is going to be that there's going to be extra genes. So here's our extra gene in between two insertion sequences. Okay, and basically what would have happened is we would have an insertion sequence that was located close to another insertion sequence, and when transposase does its job, it's going to not just get our insertion sequence, but it's going to get the gene that is in between them. And so this is where you actually get real genetic recombination happening, where we're taking a usable gene and moving it somewhere else. Maybe we only get part of a gene, and it can cause some pretty major changes in the genetic combination. So I have to stop right now or the video time is up. So if you have questions, ask, and we'll see you in class.